Hello everyone. In this class, I will discuss paleobotany topics, the types of sedimentary rocks bearing fossils, types of fossils, modes of fossilization, principle of fossils. Paleobotany. First, different types of sedimentary rocks. Generally, in majority of cases, rocks are found to may be made of minerals. Rocks may be divided into three major groups. Rocks, igneous, sedimentary, and metamorphic. Metamorphic rocks may be divided into three: igneous, sedimentary, metamorphic. Igneous rocks. These rocks are formed by the action of fire and heat inside the earth. Granite. It is a coarse granite, granite and basalt are described. It is a coarse grained rock composed essentially of quartz, feldspar, and mica. It is crystallized slowly in large masses within the crust. Such rocks are also called plutonic. Also called plutonic. And basalt. It is a dark-colored, very fine-grained rock. It is formed from flowing lava which gets quickly cooled down. Such rocks are called volcanic rocks. And sedimentary sandstone and limestone. These are these sedimentary rocks are produced by external processes from pre-existing rocks of all kinds or from organic material derived mainly from air and water. Due to heavy rain, the debris of the original rocks are carried to the sea, river mouth, etc., where they settle and gradually form the sedimentary rocks. Sandstone and shale. A piece of sandstone is seen to consist of grains of sand, identical in appearance. It is made up of fragments derived from the disintegration of some older rocks, such as granite, which trace the same minerals. It is a fragmental rock and should be powdered gently. The very fine grained sedimentary rocks corresponding to mud are called clay, mudstone or shale. Clays are soft, dull-looking rocks which can be washed by water into particles. And limestone. Limestones are accumulations of organic remains and are composed of calcium carbonate. Calcium carbonate. They efference if effervesces quickly. Efference quickly when acid is applied and give off carbon dioxide, leaving behind only a little sand or mud. Limestones are thus seem to be deposits formed from dissolved materials. The limestone is mainly composed of rounded grains that look like insect egg or sea lilies. They are made up of broken bits derived from the hard parts of animals or plants. Metamorphic rock produced on pre-existing rocks of all types.
All rocks of all kinds by internal processes. Marble, marble and crystalline limestones. Limestones are known commercially as marble when they can be effectively polished and used for decorative purposes. Limestone is chemically a very sensitive rock and readily respond to the effects of heat, pressure, and percolating waters. The term marble is restricted to the limestones which have been completely recrystallized by metamorphic processes during their burial in the earth's crust. Slate, and other is slate clay rocks pass into slates when hardened by compression. This structure is neither stratification nor lamination, but a fissure or cleavage, often of great perfection. The, the slaty cleavage depends on the orderly arrangement of minute flaky minerals such as mica, clay, chlorite within the rock. Many fine-grained rocks often split at a high angle, even at right angle, to produce a roofing slit and is called slitty cleavage. In which rocks would you find fossils and why? We find fossils in the sedimentary rock. Sedimentary rocks. Since these rocks are formed by a slow process of layer by layer deposition of sediments carried by rivers and streams into ocean and other water bodies, they are suitable for the prevention of fossils. Whereas igneous and metamorphic rocks hardly have the scope to preserve fossils. This is because igneous rocks are derived from the molten material from below the crust of the earth and are obviously not suitable for preservation of organisms at such high temperature. Metamorphic rocks, which are formed by the intensive folding and recrystallization of pre-existing rocks and the process of creating new material, tend to destroy the organic remains. That's why only in these sedimentary rocks we find fossils, not in igneous rocks, not in metamorphic rocks. And what is paleontology? What is paleontology? And what branches of paleontology? The science of fossils is known as paleontology. Invertebrate paleontology, and vertebrate paleontology. Micropaleontology, paleobotany, and palynology are the main branches of paleobotany. Fossils are remains of past life. Pseudofossils are false fossils. And basics fossils. What are fossils? And describe its various types of fossils. The word fossil is used to refer to the remains or traces of ancient life which have been presented by natural processes in the sedimentary rocks. Fossils represent the only direct evidence of ancient life. They may be found as preserved skeleton, bones, and cells of the organism, or as preserved impression of the behavior activities such as tracks, trails, footprints of the organisms. Use of the term fossil is usually restricted to the remains of organisms that died before thousands or millions of years. It is widely accepted that any organic remains found in the rocks older than 10,000 years can be termed as fossil. The organic remains younger than 10,000 years and that are still under the process of fossilization are known as subfossils. Fossils vary in size from microscopic bacteria of 1 micrometer in diameter to gigantic dinosaur or fossil tree trunks measuring several meters long. We have heard about petroleum, coal, and natural gas. These are all sources of non-renewable energy and collectively known as fossil fuels. Fossil fuels are also formed from the organic remains of ancient organisms. These contain high percentage of carbon and hydrogen and therefore are usually used as fuel. These are not considered as fuels as they provide little or no information of the past life. Sometimes coal contains well-preserved stems, leaves, roots, leaves, and cells of ancient plants, which can be identified to a certain level. Petroleum, coal, natural gas are known as fossil fuels and they are non-renewable. Non-renewable energy. They are all sources of non-renewable energies. Types of fossils. Many types of fossils are found in the layers of rocks. Among them, some are more common, such as body fossils, trace fossils, whereas some are less common. Based on the mode of preservation, size, and uses, fossils are commonly classified into many types. Based on mode of preservation, based on size, based on uses, body fossils, trace fossils. We will discuss this in briefly after writing this. Chemical fossils, pseudo pseudo fossils. They are like fake fossils, or and they are macro, large size fossils, micro, small size fossils, nano fossils, very small fossils, and uses index fossils, living fossils. First, discuss the body fossils. These are these are the preserved hard parts of actual organisms. The body means actual organisms, hard parts, body fossils. Body fossils may be the whole body of any organism. For example, skeleton of dinosaur, cell of bivalves, or body parts of an ancient animal, like bones and teeth of fishes, dinosaur, or mammals. These are direct evidence of ancient life and provide information about the shape, size, and function of a once living organism. That's why they are known as body fossils. Skeleton of a dinosaur is an example of body fossils. Trace fossils. Yeah, these are not actual remains of an organism, but are the preserved evidences on or impressions of the activities of organisms of the past. Trace fossils may include tracks, trail burrows, footprints, or borings. Trace fossils are also known as ichnofossils. Ichnofossils, and its study is called ichnology. Body fossils. The skeleton of dinosaur. Dino. So trace fossils. Footprints. Burrows. Tracks. And then chemical fossil. These are organically derived compounds that are preserved in the rocks. Generally, this compound contains no traces of the actual organism and are therefore considered as an indirect evidence of past life. It has been observed that large organic molecules do not survive long after the death of an organism, but these molecules may break down to smaller stable organic molecules which can survive over long geological time. The ancient DNA preserved in rocks is a common example of chemical fossils. The DNA preserved in rocks. Ancient DNA. The ancient DNA preserved in rocks is a common example of chemical fossils and pseudofossils. These are not fossils through they look like fossils. In nature, there are many inorganic substances which generally resemble fossils, and these are called pseudofossils. Dendrite on ore of manganese is a classic example of pseudofossils, which is often mistaken for leaves or ferns because of its tree-like structure. Dendrite is an example of pseudofossil. Macrofossils, and now based on size. They are very small fossils, less than 1 millimeter in size, and require a micro. These are micro fossils, macro and micro. Macro is large fossils. These are large fossils, more than 0.3 millimeter in size, and can be seen with naked eyes. More than 0.3 millimeter in size, and can be seen with naked eyes. Bones, teeth, cells of animal. Bones, teeth, cells of an animal, as well as hood and leaves of plant are common example of macrofossils. Hood, leaves of plants. Macro are large. Macro means large. Micro means small. Macro. Micro, these are very small, less than 1 millimeter. Less than 1 millimeter in size and required a microscope for their study. Astrocord animal fossils, pollen and spores. Pollen and spores are example. Plant fossils are common example of microfossils. Nanofossils. These are very small fossils having a size range between 5 to 60 micrometers. Micrometers. And require an electron microscope for study. Nanoplankton is a typical example of nanofossil. Nanoplankton is an example. Index fossils. These are characterized index fossils. These are characterized by short geological range, age, wide geographic distribution, rapid evolutionary rate, and distinct morphological features. For example, trilobites having a geological range from Cambrian to Permian are index fossils for the Paleozoic rocks. Index fossils are used for correlation and dating of rocks. Example, trilobites, and used for dating of rocks. Dating of rocks. Living fossils. The renowned English naturalist Charles Darwin first used this one. First used the term living fossil for the East Asian ginkgo tree. Ginkgo tree. The ginkgo biloba is the first term. This is living fossils. Living fossils include those species who have lived for long spans of geological time and are still living today with their bodies showing little or no morphological changes during the course of evolution. Living fossils have little diversity, and the actual reason behind their evolutionary stability is yet to be understood. Living fossils are very important to paleontologists as they provide the biological data which is absent in the fossils. Ginkgo to present is a living fossil of conifers. Ginkgo biloba is very important tree and in the gymnosperms. It's a living fossils. Example of living fossils. And Ginkgo biloba is used for the cytohistological zonation by Foster to explain the cytohistological zonation theory. And economic importance of Ginkgo biloba is also important, also explained. And this tree is very beautiful, beautiful yellow colored flowers. And this increases the botanical curiosity. And in next, we will study how fossils are preserved. Fossilization may occur in many ways. Sometimes the entire organism, including its soft part, is preserved. In rare cases, only hard parts of the organism are fossilized. In few cases, only imprints or traces of organisms are preserved. Hence, different organism types show different process of fossilization. Fossils occur in many different forms, such as unaltered soft parts, unaltered hard parts, altered hard part. Depending upon the nature of fossils, the process of fossilization may be classified into the following types. Or exceptional preservation, unaltered hard parts, hard parts. Per mineralization, this is further divided. Replacement, carbonization, molds and casts, tracks and trails. Unaltered soft parts. This unaltered soft part or exceptional preservation. In very rare and ideal situations, the whole of the organism, including its soft and hard parts, may be found to have been preserved. In this type of preservation, the most fragile and delicate soft parts remain unaltered and provide finer details of the soft parts. This type of preservation takes place in several ways, such as fossils preserved in amber, tar, or ice. Amber is the fossilized resin of ancient conifers. Example, amber. Amber is the fossilized resin of ancient coniferous trees. Usually, the sweetness of resin attracts the insect and they come in contact with resin. Due to the stickiness of resin, the insect get trapped in it. Later, lumps of resin fall on the ground and get buried in the sediment and finally turn into solid amber. The Western lignite mine in Surat district of Gujarat yields fossiliferous amber with well preserved insect. Some organisms may fall into crevasses in glaciated areas or become trapped in permanently frozen soil and thus get preserved perfectly. Low temperature and aerobic without oxygen condition in frozen area allow the best preservation of the soft body parts of any organism. The woolly mammoth, preserved in the Pleistocene sequence of Siberia, is a classical example of preservation in frozen ice. The examples are amber, the western lignite in Surat, district of Gujarat, yields fossiliferous amber with well preserved insects. And some other, the woolly mammoth fully mammoth preserved in the Pleistocene sequence of of Siberia is a classical example of frozen ice. Preservation in preservation in frozen ice. Sudden burial of ecosystem by volcanic ash and sandstorms also leads to excellent preservation of fossils. The well-preserved Cretaceous biota of China and Mongolia are well-known examples of biota quickly buried by volcanic ash and sandstorms. These are unaltered soft parts or exceptional preservation. And after that, these are altered, unaltered soft parts. Unaltered. These are altered hard parts. Preservation. Many invertebrates possess hard parts made up of stable compounds such as calcium carbonate, calcium phosphate, and silica. It has been found that many cells and skeletons of invertebrates were preserved in the sedimentary rocks with no alteration in their hard parts. However, it does not mean that that the organisms are unchanged. In fact, the less stable organic material, soft parts, is removed from their bodies during the process of fossilization. Fossil record has numerous examples of fossil cells which are millions of years old yet cannot be easily differentiated from the modern cells. Altered hard parts preservation. These are skeletons preserved in skeletons cells of invertebrates were preserved in sedimentary rocks with no alternation in their hard parts. However, it does not mean that their soft parts are removed from their bodies during the process of fossilization. Now, altered hard part. Altered hard parts. The story of alternation of hard parts starts after the burial of the organism. Most cells and skeletons of animals contain pores. For for example, bone marrow in vertebrates. Once an organism gets buried under the thick cover of sediment, the water containing dissolved minerals present within the sediment fills the porous and replaces or alters the hard part. The pressure generated by sediments facilitates the process of alteration, which may take place in following ways. Permineralization. This petrification or permineralization it is a slow process and involves the removal of organic material by mineralized solution. Most of the cells, bones, and wood are porous. In addition, the decay of soft parts increases the porosity of this organic material. When these porous materials are buried under sediments, the overburden increases the temperature and pressure. As a consequence, the chemical reaction occurs between groundwater and soluble minerals present in the sediments. This reaction leads to the formation of mineralized solution, which fills the pores of organic material, thus having the way for the solution to precipitate in pores. In this way, the organic material is partially or completely replaced by the inorganic matter, consisting of silica, calcite, or pyrite. Petrification is a very slow process in which replacement occurs molecule by molecule. As a result, very fine details about the organism get preserved. The petrified wood of tree and bones of dinosaur or tusks of elephant are common example for mineralization. Example: wood of tree, bones of dinosaur, and tusks of elephant. Tusks of elephants are petrification examples. Replacement. It is also a slow process of fossilization. In this process, the underground water slowly fills the pores of organic material and completely dissolves the hard parts of the organism trapped in sediments. At the same time, the groundwater carries minerals in solution that replace the organic material with such minerals as silica, calcite, hematite, and pyrite. And carbonization. Third is carbonization. Carbonization. In this process, the soft-bodied animals as well as the stem and leaves of plants buried under the sediments are decomposed or pressed and thus losing hydrogen, nitrogen, oxygen and other volatile constituents. As a result, a thin carbon film is left behind. Such films are enriched in carbon and appear as black shiny fossils. Carbon films create impression in the rock, outlining the shape of the organism. Plant leaves are commonly preserved in this process. We may find most of the carbonized fossils in the coal mines. Plant leaves. Example for this. And the last, then molds and cast. Molds and casts. Molds and casts. This process involves the natural duplication of the original organism. When a cell gets buried in sediment, subsequently the sediment becomes hard. At the time of the water running through the sediment may dissolve the cell completely, leaving behind an impression or a void of the cell. The such an impression is known as mold. If the void is filled with grain or of sand or clay, it hardens and produces a replica of the original cell shape, which is known as cast. Mold, cast, replica. It's a replica, and it's an impression. Tracks and trails. While moving on the soft ground, such as mud and sand, the organisms may leave behind impressions of their movement. For example, the footprints, burrows, and boardings are preserved when the soft ground material harden and in rock. This type of fossils impression are also described as trace fossils. Footprint, burrows, and the importance. Importance. of fossils in geology. Importance of fossils in geology. Geology is the science dealing with the study of planet Earth, and this study can never be completed without taking fossils into consideration. We know, fossils are naturally occurring clues to the past life and therefore are important to interpret the history of Earth as well as evolution of life on it. As a result, fossils are often considered as written texts preserved in the rocks in the form of remains of ancient life that provide valuable clues to the following: evolution of life. This provide evolution of life. Organic evolution is a sequence that connects organisms to one another through geological ages from an evolutionary point of view. Further, it establishes ancestor-descendant relationship. The theory of organic evolution states that today's living more advanced and complex life forms had evolved from the more primitive and simpler life forms that lived in the geological past. It should be noted that fossils strongly and clearly support the idea of organic evolution. The fossils of the most primitive life forms like bacteria, blue-green algae, or Ediacara fauna are found in geologically older rocks. For example, rocks of Precambrian and Lower Paleozoic. Precambrian and Lower Paleozoic. However, fossils of more advanced life forms like vascular plants, mammals, or birds occur in geologically younger rocks. For instance, of Phanerozoic times. As the life has changed throughout the geological time and the rocks of the same geological ages contain similar fossils and rocks with different ages have different fossils. Based on the changes in life, the history of Earth has been divided into five main times division which form the geological time scale. This division from oldest to youngest are Cenozoic. It is marked by the presence of recent or modern life. Cenozoic and then Mesozoic. Mesozoic. It is marked by the presence of medieval or middle life. Paleozoic. It is marked by the presence of ancient and or old life. Proterozoic. Pro, protero, Proterozoic. And it is marked by the presence of earlier life. Hmm? Archaeozoic. Archaeozoic. It is marked by the absence of life or presence of most primitive life. The above mentioned sequence of life clearly shows that life has gradually changed from simpler to simpler forms to more complex forms. It is thus true to say that the study of evolution of life forms through geological age and can only be possible by making a detailed and systematic study of fossils. Prehistoric life. When we talk of prehistoric life, it refers to the life of the past that lies between first appearances of life, about 3.8 million, 3.8 years ago, million or 3.8 ago, billions ago, to the first human written record or document of history, some 10,000 years ago. Fossils are the only evidence of the past life, and many of them represent once living organisms that are now extinct. As mentioned earlier, life has changed gradually over a long span of geologic time by the process of organic evolution. In addition to organic evolution, other factors such as predation, disease, and competition with other organisms may also cause the disappearance or extinction of some organisms. Such organisms are left with no living representative. The fossils are only a valuable source of information about the extinct organism as well as biodiversity of the past. The gigantic dinosaur, ammonites, trilobites, and woolly mammoths are examples of some extinct animals that are known today only by their fossil record. The gigantic dinosaurs, ammonites, trilobites, trilobites, and woolly mammoth. Woolly mammoths. Dating of rocks. It helps in dating of rock. Dating of rocks. Life is continuously changing through time, resulting in the appearance of new life forms and disappearance of some of the older ones. The sedimentary rocks contain fossils deposited layer by layer. The principle of order of superposition states that in a sedimentary rock section, the layers at the bottom are deposited first and hence are the oldest. The beds overlying them are younger. The fossils contained in this succession will keep changing and it may not be possible to have the same forms throughout the succession. As a result, it is possible to characterize the different layers of rock succession based on their enclosed fossil. We assign relative geologic age to this fossiliferous fossiliferous layers on the basis of their fossil content. The branch of stratigraphy that deals with relative ages of sedimentary rocks is known as biostratigraphy. Biostratigraphy. There are some ancient organisms which lived on the earth for a very short period of time, but they had a wide geographic distribution, rapid evolutionary rate, and distinct morphological features. The fossils of such organisms are called index fossils. And these have served as key fossils to date the rocks. For example, trilobites are used as index for dating rocks of lower Paleozoic erythrum, and ammonites are used for dating the rocks of Mesozoic erythrum. Fossils are also used for biostratigraphic correlation, in which rock successions of two geographically separated areas can be correlated on the basis of fossils present in them in order to understand their spatial and temporal distribution. Paleogeography. We may be surprised to know that geographic distribution or position of land and sea was very different in the geological past than what we have today. The study of past distribution of land and water is known as paleogeography. The fossils present in the layer of rock succession help us to reconstruct the past distribution of land and water. It is a well known fact that India was once a part of the former Gondwana continent, which separated from it during the Mesozoic era and collided with Asia in the early Cenozoic era. The present day Himalaya was once upon a time a sea, and horizon between Indian peninsula and Asia is marked by the formation of the great Himalayan mountain system, whereby the sea bottom got uplifted. It is the study of fossils recovered from the Himalayan mountain that tells us about the existence of Himalayan sea that got finally uplifted into a mountain range. In the Cenozoic era, based on the fossil similarities between South America, Africa, Australia, and Antarctica and India, we know that these landmasses were united during the late Paleozoic era and formed a supercontinent known as Gondwana landmass. Climate. Certain organisms, particularly paleoclimate, certain organisms, particularly animals and plants, are highly dependent on environmental conditions, and many of them are narrowly adapted to specific climatic conditions. As a consequence, their fossils provide valuable clues to know the climate of the past or paleoclimate. Interpretation of paleoclimate from fossils involves knowledge of the relationship between present day distribution of organisms and their climatic conditions. For example, the fossils of reptiles such as lizards, snakes, etc., are indicators of a warm paleoclimate because they cannot live in cold climatic conditions as their body is not able to maintain constant warm temperatures. The fossils of the plant cycads indicate tropical and subtropical ancient climate. Because modern cycads occur in these climatic zones, the margin of plant leaves are excellent indicators of past climate. For example, fossil leaf with smooth margin are good indicators of tropical paleoclimate, whereas leaf with toothed or lobed margins indicate cold climate of the past. In paleo environment, paleo environment. Like paleoclimate, fossils are also good indicator of ancient environment. That is paleo environment based on the fossils. It is easy to recognize marine and non-marine environments. The marine paleo environment is characterized by the presence of cyanobacteria, foraminifers, radiolarians, corals, brachiopods, bryozoans, some bivalves, ammonites, and trilobites. However, the presence of fossils of plants, fungi, and some gastropods and astropods, insects, amphibians, birds, and most mammals in the sedimentary sequences generally indicate non-marine freshwater and terrestrial paleo environments. Discovering oil and discovering oil and coal deposits. The oil and coal deposits are the products of ancient animals and plants. Hence, fossils play a key role for exploring them. Both these deposits are the products of ancient animals and plants. Both these deposits are located deep underground. In petroleum exploration, fossils are used to describe and predict the temporal and spatial distribution of source rock, reservoir rock, as well as cap rock. Fossils are also used to determine the precise age of petroliferous basins and correlation of oil wells and boreholes. During oil exploration and drilling for oil and gas, it is necessary to take precise correlation of the oil wells to determine hydrocarbon reserves. This is easily done by the use of microfossils obtained from the core samples and the process is called well logging. For core exploration, the plant microfossils such as spores are useful to identify and correlate the coal seams. Three microfossils groups, namely foraminifers, calcareous, nanofossils, and polynomorphs, pollens, and spores are commonly used for correlation of oil and coal deposits. And some key principles of paleobotany. Principles of paleobotany. Principles of paleobotany. Uniformitarianism, interpreting ancient plants based on the process observed in living plants today. Example, assuming ancient plants had similar environmental requirement to their modern relatives. Principle of succession. Specific plant fossils are characteristic of a specific geological time period, allowing scientists to date rock layers biostratigraphy. Correlation. Similar fossil plant assemblages found in different geological area indicate similar geological ages. Reconstruction and morphology. Reconstruction. Reconstructing entire plant forms from fragmented fossils, stem, leaves, and pollen to understand structural evolution. Paleoecological context. Reconstructing in ancient ecosystem of and inferring paleoclimatic climates based on plant adaptation such as leaf size, stomatal density, or food structure. What are plant fossils? The main modes of preservation. Plant fossils are the remains of the past life that have been preserved in sediments of the earth crust. The study of fossils plants fall into two disciplines, paleobotany and palynology. Plant fossils divide into tiny and palynology. Wood, flowers, leaves, seed, petrified wood, petrified wood, flowers, seeds, leaves are studied in paleobotany. In palynology, such as pollen and spores are studies microscope. These are macroscopic and these are microscopic. Palynology is usually treated as a branch of micropaleontology. The mode of preservation. The fate of most organic materials material produced by living system is to be decomposed to carbon dioxide and water and recycled into the biosphere. The fact that organic matter is present in many sedimentary rocks indicate that the decomposition is not 100% and plants and other organic remains get preserved as fossils. Plant become fossilized in a variety of ways. Each type of preservation carries different information about the one living organism. Also, not all organic compounds are equally resistant to degradation and decay. Plant cell walls are composed primarily of the polysaccharide polymers cellulose and are far far more likely to escape decomposition than internal membranes and organelles, so which are rich in protein, lipids, and sugars. Secondary compounds such as those impregnating or covering cell walls are also resistant to decomposition, such as lignin, wax, cutin, and sporopollenin, which forms the external cell of spores and pollen. For the preservation of plant fossils, these three conditions are required: rapid burial in sediment, conditions for preservation. Rapid burial in sediments, reducing burial environments, reducing burial environment, that is absence of oxygen, that's absence of oxygen, fixing the organic material to retard anaerobic decay. Consequently, plant fossils are well preserved in environments that are very low in oxygen because most decomposers require oxygen for metabolism. Such sediments are commonly gray, green, or black in color. Plant fossils are usually found in fine grained sediments such as sand, silt, or clay, and also in association with organic deposits such as peat, coal. Six modes of preservation of plant fossils are commonly recognized and are listed below: compression, impression, cast and molds, permineralization, compaction, and molecular fossils. Compression. Compensations are plant remains that have suffered physical deformation, such as that the three-dimensional plant parts are compressed into two dimensions. Compression preserves organic matter such as leaves that often retain cuticles. Peat too is a compression, a thick accumulation of plant debris relatively free of mineral sediment. Compressions are excellent records of external form, especially for planar structures like leaves. Impressions. Impressions like compressions are two-dimensional imprints of plant found most commonly in fine-grained sediment such as silt or clay. However, unlike compressions, they are completely devoid of any organic matter. In fact, impressions are compressions without organic material. The impressions of plants preserved in the fine-grained sediments contain remarkable details of original external form of plants. Impressions like compression record information about external shape and morphology of plant organs. Leaves are among the most common organs preserved as impressions. Let's try to think what is the type of preservation in a rock which was split apart was found to have plant imprints on its two slabs. The organic matter, however, was adhered to only one side of the rock. In this case, the side with the organic material is the compression, while the corresponding side without the organic material is impression. The compression is the side with the organic material is compression, with the corresponding side without the organic material is impression. Third is casts and molds. When sediments is deposited into cavities left by the decay of plant parts, a cast is resulted. A mold is essentially a cavity left in the sediment by the decayed plant tissue. Casts and molds usually lack organic matter. Casts and molds may be found together with the cast filling the mold. Molds are found when soft sediment surrounding the structure lithifies or hardens. Before the structure decays, when the mold gets filled with sediment that subsequently hardens, a cast is formed. Casts and molds record external or sometimes internal organ feature. Unlike compression and impression, molds and casts often are true records of the original three-dimensional shape of the plant structure. These are three-dimensional. Two-dimensional. Two-dimensional with organic material. Without organic material. Per mineralization. It occurs when the plant tissues are infiltrated with minerals rich fluid. Minerals like silica, calcium carbonate, phosphate, or pyrite precipitate in cell and intercellular spaces, thus preserving internal structures of plant parts in three dimensions. This type of preservation is also known as the structural preservation of organic material, and thus permeation yield detailed information about the internal structure of the once living plant. The mineral matter many times replaces the cell wall or and other internal structure wherein the organic material is lost. Such kind of preservation is called petrification. The mineralized wood thus preserves the cellular detail of food anatomy and lignin of the cell walls, that is organic matter, by mineral infilling, whereas petrified wood, on the other hand, lacks cellular preservation and organic matter. Silica permineralized silification occurs in areas where silica-rich volcanic plastic sediments are weathering, such as famous upright trees of Yellowstone National Park, USA, whereas permeation with calcium carbonate, calcite, or dolomite is particularly common in coal seams, where peat gets mineralized as coal balls, such as Carboniferous coal seams. Coal balls are rounded to ellipsoidal fossils that preserve plant remains. Coal balls. Compactions. In compaction, plant retain their external form with only slight volume reduction. That is, it is three-dimensional compactions, possesses organic material, and are not mineralized compactions. No, not mineralized. Compactions are common in peat, down coals, lignite, and soft sediments, and more so if the fossils are younger. Pollen and spores are often preserved as compactions. Internal structure, especially of thick-walled, hard fruits, is sometimes well preserved as compactions. Molecular fossils. Thick-walled, hard fruits break down products of chlorophyll, carbohydrate, and lignins preserved in fossil leaves, and lipids and their derivatives in sediments are the example of molecular fossils. Molecules of oleaginous formed by flowering plants, some ferns and lichens, are more usually found preserved in sediments. Rarely, but genetic material, DNA and RNA too, get preserved. The preservation of these chemical products of plant is highly variable. Further, it depends on oxygen levels during deposition and different temperatures experienced by the rocks since preservation. Plant fossils are very useful in geology. Plant fossils are significant in many areas of geology. They have played a key role in correlating sedimentary sequences, reconstructing past geography, climate, and ecology, in addition to understanding plant evolution on the Earth. Let us discuss some important utilities of the plant fossils. Correlation of rock units which are widely separated, geographically based on similar fossil leaves, seeds, etc. Fossil plants have been used in paleogeographic reconstruction, that is reconstruction of the paleo position of continents with respect to each other. Recall the presence of the class of trees flora during the Carboniferous Permian times in South America, Africa, India, Antarctica, and Australia, as discussed. This plant has been cited as evidence for the formation of landmasses into a supercontinent known as Gondwana land. Paleoecology. The study of the past ecology can also be deduced Paleoecology. The study of past ecology can also be deduced from the fossils. The principal means of inferring paleohabitat is through the assumptions that habitat preferred by modern analogues is that of fossil relative. Using the uniformitarianism principle is the key to the past. That is present is the key to the past. The area from where fossils plants are recovered, its paleoecology can be reconstructed. An important application of plant fossils is determining the paleoclimate, that is the climate of the past. In this regard, several approaches have been made over the years. A brief account of which is given here. A nearest living relative NLR method is based on the premise that climate preferences of the fossil are similar to those of the NLR. Thus, in order to infer past climates, comparison of as many fossils as possible within a flora of their most closely related living taxa is made. The more species in a fossil flora that have NLRs, the more precise is the paleoclimatic interpretation. Also, more clearly, a fossil taxon is related to an extent one. More precise is the result. The method is particularly useful when dealing with Cenozoic floras, as they are more likely to have close living relatives further back in time. The method is less effective as older fossils taxa do not have close living relatives. Upchurch and Wolfe in established ways of accessing paleo temperatures and rainfall measures based upon the plant morphology, drawing analogy with the modern ones as largest leaves are found in tropical rainforest and size diminishes as temperature and moisture decline. In tropical areas, leaves have entire unbroken margins, whereas in temperate areas, there are many more leaves with toothed margins. The upchurch, upchurch and Wolfe established ways of accessing paleo temperatures and rainfall measures based upon the plant morphology, drawing analogy with the modern ones as largest leaves are found in tropical rainforest, and size diminishes as temperature and moisture decline. In tropical areas, leaves have entire unbroken margins, whereas in temperate areas, there are many more leaves with toothed, toothed margins, like needle type, toothed margins. Leaves from tropical rainforest species have elongated tips to allow water to clear the leaf during excessive downpours. The proportion of deciduous trees, those that shed all their leaves simultaneously in winter or during the dry season, to evergreen is highest in temperature ones. Dendrochronology on tree ring dating is a method of dating based on the analysis of patterns of growth of tree growth rings. Dendrochronology or tree ring dating. It is tree growth ring. It is used to determine the age of archaeological sites by studying the growth rings present in the wood that has been used for construction, paintings, and furniture, etc. In addition, the fossilized plant organic material serves as a sample for carbon dating as well as for stable isotope studies for determination of past levels of salinity, temperature, and carbon dioxide content. The techniques now now techniques involved in the study of plant fossils. Techniques involved in the study of plant fossils. Scientific Scientific techniques. Radio carbon dating. Radiocarbon dating is a method that provides fossils age estimates for carbon-based materials that originated from living organisms. The method was developed by an American chemist Willard Libby, Willard Libby in late 1940s, and and soon become a standard tool for archaeologists. Libby received the Nobel Prize in chemistry for his work in 1960. The radiocarbon dating method is based on the fact that radiocarbon is constantly being created in the atmosphere by the interaction of cosmic rays with atmospheric nitrogen. The resulting radiocarbon combines with atmospheric oxygen to form radioactive carbon dioxide, which is incorporated into plants during photosynthesis. Will animals get this carbon by eating the plants? And the other is 14C carbon, ground thin section. Ground thin section technique. Ground thin section technique. This technique is suitable for the study of petrified fossils, which preserve cellular details. In this technique, the specimen is cut into small sized sections and surfaces are smoothed. The smooth surface of this section is mounted on a glass slide. Now it is warmed and coated with melted resin. Now the specimen is cut as close to the glass as possible, thus a moderately thin section is obtained. The thin section fixed to the glass slide is further ground on a revolving lathe with 100 card cardboard. The till sufficiently translucent so that the section can be viewed under the microscope. Finally, the slice is mounted with a cover glass using suitable mounting media. Advantage are a thin section of petrified wood can be made that preserves the cellular structure in an unchanged condition. Disadvantage is that this technique have the following disadvantages, like the technique need extensive labor and is time consuming. Advantage: cellular details, clear cellular details. And disadvantage: advantage disadvantage. The technique is laborious and time consuming. And second is, a small number of sections can be made from a given specimen. Thus a lot of material is wasted. Lot of material is wasted. Peel section technique. Peel section technique. This is a conventional peel section technique. This is a conventional and most suitable technique for the study of well-preserved petrification with considerable organic material. In this technique, the etching of the fossil surface is done with the help of some mineral acids. Example: 5% hydrochloric acid if the material has calcium carbonate, and 10% hydrofluoric acid if the material has silica for 10, 5 and 10 minutes respectively. The etched surface is gently passed under running water for the removal of acid and is air dried and then covered with a solution as a thin film of nitrocellulose. Advantage: a series of sections can be made from a single specimen. It is less expensive and quicker to prepare. Advantage are less expensive and quicker to prepare. The sections peels obtained and translucent, thinner and durable. And fourth is transfer technique. It is an important technique for the study of qualified compression. This technique is most suitable for the study as it reveals additional details of venation, epidermal pattern, and hairs. In this technique, the peel solution is coated in the delicate fossil material adjoining the rock surface. When solution dries, the portion of the rock having fossil material is removed. The face of the specimen adjoining the rock surface is cleared either mechanically and or by washing in an acid for removal of rock particles. The prepared surface of the rock is coated with a solution of nitrocellulose or with a cellulose acetate film. When the film is dried, in exine, pollens and spores are considerably preserved. When film is dried, it is loosened from the rock surface. Sometimes qualified materials are adhered to the film. Occasionally, the film is treated with a strong oxidizing agent to make the film more transparent. Finally, the film is dried and permanently mounted on the slide with a cover glass using a suitable mounting medium. Advantages are very useful for the study of qualified compression. Useful to study qualified compressions. It helps to learn about leaf forms, venation pattern, stomatal and epidermal characteristics, which are important features used in establishing systematic and phylogeny of extant plants. And last is macration technique. Technique that this technique is most suitable for the study of peat, lignite, and coal. It is very useful for pollen and spore analysis. The advantage are a small portion of sediment is enough to get considerable amount of pollen and spores. Due to the presence of resistant chemical sporopollenin in exine, pollens and spores are considerably preserved in deposits like peat, coal, lignite, etc. The macration process is very simple and reliable. This is advantage is very simple and reliable. And small portion is enough to get considerable amount of pollen and spores. And modern techniques. Nowadays, fossils are studied by modern techniques using transmission and scanning, electron microscope, interference microscope, phase contrast microscope, and by X-ray analysis, X-ray and CT scans. Researchers can study the anatomy of fossils without damaging the material by using X-ray analysis, radiography, and CT scans using computed tomography. which provide detailed internal images of fossils. Fossils. The paleobotanical nomenclature. Paleobotanical nomenclature. Dead plant remains rarely get fossilized as they are liable to get disintegrated. Softer plant parts have greater chance of early disintegration, and the harder plant parts have the greater chance of fossilization as the entire plant cannot get fossilized. Hence, fossils of only portions of such plants are found. After that, naming of those portions or plant parts are given under some form genus or artificial genus, which does not indicate actually to what plant that portion belongs. Each of these names acquires the status of a genus. Example of form genera are Stegmaria, rhizophore of Lepidodendroid, Lepidotropus, strobili of Lepidodendroid, etc. In naming such form genera of fossil, generally fish suffixes signifying which plant part it represents are taken into consideration. Thus, phylum means a leaf. Example, Tylophyllum, Nymphophylum, etc. Pteris is a fern-like stem or frond. Example, Lygenopteris, Sphenopteris, Archaeopteris, etc. Dendron is considered as a trunk of a tree. Example, Lepidodendron. Xylem is a woody part of the plant. Example, Chloxylon, Myelosylum, Dedoxylum. Spermum, example, Coryspermum, Carpus. Example, Trigonocarpus, Cardiacarpus. Carpon, example, Mosocarpon, Lepidocarpon. Stoma, example, Legionostoma, are considered as seeds. Theca, example, Chrosotheca, is the microsporangium. Philostropus, example, Lepidostropus, Androstrobus signifies a core of prehistoric plant. Sometimes inflorescence signifies as thus Cordian, Cordiana, Cordiana, thus. So it is the work of the paleobotanist to collect portion of such form genera to reconstruct the full plant as it existed in the prehistoric ages. Example, Williamsonia sewardiana. After Birbal Sahani. Such work is surely very tough and sometimes may even lead to wrong opinions. Another example is cited here. It is the name of the plant, fossil plant, Calimotheca housi, Lygenopteris oldhamia, which bears a root called Holoxyron hookeri, stem known as Lygenodendron, petiole as Sphenopteris hoinopausi, rachis as Rachopteris aspera, microsporangium known as Telangium Chrosotheca, and the seed as Leginostoma lomaxin. The complete plant was reconstructed by careful placing of the plant portions together as the plant was not found as a whole in fossilized condition. Phylum, leaf. Phylum means leaf. Pteris. Pteris is for like stem or front. Dendron, dendron is trunk. Dendron is a trunk. Xylon is a woody part of plant. Woody part of plant. Spermum. Spermum carpon. Stoma are considered seeds. Theca is microsporangium. Strobus is cone of a prehistoric plant. Inflorescence and the fossil plant is example of fossil plant Laginopteris. Laginopteris oldhamia. Kelly Meto Thika Warning Housing Laginopteris oldhamia. Root Kedo Xylon Hook Kerry Stem Digenodendron Petiole Spinoteris Hoining Housing. Lycopteris aspera Microsporangia Telangium Crosso Theca and seed as Laginostoma lomaxi. This is how the Laginopteris oldhamia classified, was reconstructed by the careful placing of the plant portions together, as the plant was not found as a whole in fossilized condition. By part, the reconstruction was done when mm, then they don't know the relations between this, that these are the parts of same plant. Root, stem, petiole, when they did not know the root and stem of these are of Laginopteris, and after that mm, it is named as Laginopteris oldhamia. This is this is of same plant. Root is Caloxylon hookeri, stem is Ligonodendron, petiole is Spinoteris housing, Rachis Rachu, Teres Aspera, Microsporangia Prosothica, Terengium, also known as Terengium Prosothica. Seed is Laginostomal lomaxin. Is the nomenclature is completed. The phylum leaf, teres, fern-like, dendron, stem, trunk, xylem, woody part of the plant, sperma, seed, theca, microsporangium, strobilus, cone of a prehistoric plant. Thus, inflorescence is like trace of techniques, radiocarbon dating, Willard Libby in 1940 Nobel Prize in Chemistry for this radiocarbon dating, and he was chemist in chemistry 1960. Ground thin section technique, peel section technique, transfer technique, and migration technique in modern techniques. The CT scan, X-ray, the conditions, laboratories, the plant fossils. Thank you everyone. I will meet you in the next class.